And I hope that's okay with you. I can speak Danish, but I'm not used to talking about maths stuff in Danish, only in my own area of expertise. So I'm going to talk to you in English. And I'm going to try and talk to you about the relevance of maths in the bio area. And Carsten has asked me to talk a little bit about integration with multiple variables and stuff like that, and a little bit of an introduction into what's coming up next in your course, and I'll also touch briefly on that. But I'm going to start to talk a little bit about myself. I come from Australia. I actually have a Bachelor of Science with honours, which is a bit like a 50-point exams project in Denmark, and a PhD in the fermentation area. So I'm not a maths person or a chemical engineering person or a hardcore calculation type person, modeling type person. And all through I did maths at first year at university because I thought, you know, it's a bit like medicine. It tastes bad, but it's probably good for you. And I did maths at first year, and I was very glad to do that. But I didn't think I'd really be using maths very much because I was much more interested in the bio side of things, microbiology in particular, and biotechnology. And doing a Bachelor of Science, you sort of didn't come into the maths side of stuff very much. If I had have done chemical engineering, it would have been a different thing. But nevertheless, I found that I've been using more and more maths. And this is an example of my latest paper that's just been submitted for publication. And as you can see, it's all about modeling stuff, actually. So a biochemically structured model for ethanol fermentation by Clivoromyces maxianus, a yeast, batch fermentation and kinetic study. And that's me there. And there's some people from chemical engineering and some people from Italy involved as well. So I've come actually quite a long way from just sort of playing around with agar plates and looking at the way things grow to coming to actually using mathematics a lot. And I'm glad for my basic knowledge of maths, but I would actually really like to have a better knowledge of maths. You find that maths comes into bio in all over the place, in fact. And this is a little bit more from that same research paper. And this is a model of a cell, or a model of a bioreactor, in fact. And you have the substrate coming in the top, lactose, and it's turned into some products. And the product we're interested in is ethanol, but there can be byproducts, and the cells multiply, get more cells as well. And you can look at it as a black box and look at the rates that these things are produced, and you can develop a model based on that. Or you can actually start to look at what's going on inside the cell. And that's what you see here. We have a very simple structure to the metabolic pathways inside the cell. So there are a number of reactions going on. It's very simplified here. Where the substrate comes in, it's converted to different things inside the cell, which can end up being different products which come out. And you can represent this structure with this set of equations here. So this is the model that's coming out of this simple structure of the cell. So this is a very important way to look at what happens during a fermentation. We want to get more ethanol. We want to understand, why do we get glycerol? Why do we get acetaldehyde? How do we get more of this? How can you manipulate this? How can you change the process? And by creating a model, that's the way you can do this. You can predict the way you can get more of your product. My interests are in fermentation, and this is a little bit dark, I can see. It looks fantastic, of course, on my computer, but it's a little bit dark here. My interests are in fermentation area and downstream processing. And what's not so obvious here, maybe you can look at it afterwards on the video, and it might come up a little bit clearer. But what you see here is actually a pilot scale fermenter, 200-liter fermenter, and one of my PhD students has been using that. It's a new type of fermenter, developed actually in Denmark by Isomix, which was bought by Alfa Laval now, and they've developed a new type of fermentation system. And this has been studied for fermentations, but also to look at mass transfer, mixing, heat transfer, all sorts of things that go on inside the fermenter. And this is a giant chromatography column. It looks a bit like a spa in reality. It's 1.5 meters in diameter, so it really is big. And this is a special type of fluidized bed chromatography, where you pump the liquid in the bottom and you're able to separate and purify proteins, etc. So I've worked in these types of areas, and I find this really, really fascinating. I come from Australia, as I just mentioned, and what's coming up in the course, maybe you've already started to hit on it, are some examples. And I was very interested to see what Carsten sent to me, that you have probably an example on bushfires. And that's very, very relevant for me coming from Australia, because you have these really, really horrendous bushfires. And I was quite interested to see that you have an example on how you can model the progression of these bushfires. So I'm not going to talk about this because you will be introduced to that at some other time. But when I was sort of thinking around how do bushfires move and how are you modeling things and stuff like that, and I looked around in the literature, and I've taken a lot of sort of examples here from the internet and literature and stuff like that, then I was interested to see maybe there are some parallels with the way cells move. And that's what we have here. We have a cluster of cells, and this is taken from this paper here, so you can go and look it up later on. You can go into Portalen, go to the library, Biologi, and then you can go to the article database system, and you can find this paper, and you can download it and have a look if you like. But here we have a cluster of cells from this weird thing called dictyostelium here, which is like a multicellular organism, and it says here a slug of dictyostelium, so a snail, a dictyostelium here. And it's composed of a whole heap of cells, and they are actually modeling the way these cells move. And they've introduced some, like, what are they called here? Pacemaker cells. They've introduced pacemaker cells here and here, and that's why you see this moving off the side here. And they have developed a quite sophisticated model to understand what's going on here and why the cells move. And this can have a wide-ranging repercussions also in the human body. How do the cells move? How do wounds heal? Etc., etc. So I think you've heard a hell of a lot about vectors, and you're also going to hear more about vectors in these examples when you come into your integration in many variables and things like that. So I'm not going to spend a lot of time. This is stuff I pull off the internet, and for me I thought it was kind of interesting to get a little bit of a refresher on maths. And so go in and have a look about it, but maybe Carsten will present it in a bit more of a sophisticated, knowledgeable fashion to me. But anyway, you know all about vectors, and vectors can move in different ways. They can come out of the page, and you can have them in 3 dimensions and 2 dimensions, etc., etc. You can start to model the way that the vectors move away from a point in the center, or the way they move into an area, diverging or converging vectors and flow. You can also begin to model, if the flow is moving around, the vectors that are responsible or can describe that movement. And here we have something in the center, and it's rotating, and you have these vectors which can describe the movement. And then you can start to say, okay, what happens if we're not just revolving around this point here, but we also have something that's also revolving in this flow field. So you can start to develop your description of the phenomena. So here we have this one rotating around this x, here rotating around, sorry, I think this is the y here, the x, and the z here. And you can start to see that, well, you can model complex flows, bushfires, movement in liquids, etc., using these kinds of vector systems. You can look at 2 dimensions. So here we have a flow around like this, and you have a whole lot of little microflows inside it. And you can also model structures or surfaces a bit like this. If you go into this homepage, then you can play around with this surface. You can pull it from all different directions. It has a lot of nice animations, actually. You can also model more complex surfaces like this surface area. And one of the things that you're going to come up to hear more about is flux. And flux is, for example, moving through a surface, a flow or air or liquid or whatever that's moving through a complex surface. So you have a whole description here with all of the arrows showing the movement of this flow. And here you have the actual surface, and you can break it down into small areas, and you can model the flow of liquid through a surface. And you're going to come into more of this about when you look at the bushfires and you look at the way more complex flows and things move. So I'm not going to spend a lot more time. I'm going to come back to this and say we saw this example here with the cells moving. And in this paper, then they have then started to simulate the way the individual cells move. So this is a cell track simulation of each of the cells here. So you can see we have the different cells, and they have the little tracks following where the cells have been moving as they've gone from each frame to each frame over time. And you can easily see that looking at the way these cells move, that you could set vectors to those, and you could start to model the way the cells move, just like in the examples that I've shown you. But the interesting thing is that was looking at the cell movement from this sort of perspective of the cells themselves. How do they move? Looking at them. But then you can start to try and make the model even more sophisticated. Remember I talked about the black box model of the cell at the start of my talk. You can look at the cell from the outside or the fermenter from the outside, but then you can start to look into the structure to try and understand it even better. And that's what these people have done. It's from the same paper. And they've started to look at why do cells move the way they move? What is the mechanism underneath that? And try to understand and develop their model, extend it even more. So they have an area which elongates here, shown by the arrow. It elongates out, and it sort of stops, grabs fast, or grabs the surface here, and sort of pulls the back end along. So it extends out, elongates out, grabs hold, and then pulls the back end along. That's the way the cells are moving. And then when you have a number of cells present, so it's not just a linear two-dimensional flat surface, you have a heap of cells in 3 dimensions. How do they move and how do they interact with each other? That is also modeled here. So they've taken the model to the next level. And you can say, okay, these cells move incredibly slow, right? But still, you can model them with vectors. You need to look at it in 2 dimensions and in 3 dimensions. But these guys, or actually not these guys, another group from another paper, they extend this model even further, the model of cell movement. Why is it and how is it that the cell is able to push the front bit out and pull the back bit along? So this model, it's now been extended. You have the surface of the cell here, the front of the cell being pushed out, and here they have reconstructed, made a model of the mechanism inside the cell which is responsible for pushing it forward. All of the different substrates that are used to build up this scaffold and push it forward. Calculated the angles and everything like that, how many of the different compounds are involved, etc., etc. And they have developed this model. You can see sort of a vector field here, just like what you will be looking at in your bushfire and your other calculus examples. And here they have a picture to sort of verify this is what's going on, where they have dyed the different parts of the structure, and they can see that the way the cell actually moves. So they've verified the model. So you might think, okay, yeah, can you really model cells and the way they move? And can you describe it mathematically? And you can. You can describe it mathematically on a whole lot of different levels. Looking at the cells, the individual cells, the way they move, looking at the way they interact with each other in 3 dimensions, and then going inside the cell and modeling the structure and the mechanisms inside the cell. Another example which I came up with was this one here. And here we have, it's a model about cell attachment, and they're looking at the way that genes get transferred in the environment by cells. And you can go into this paper and have a look. It's quite a recent one from 2010. And they have developed a model here. You can see the model with these different spheres here. And they have a surface on this model, which you can see represented here. And what they are looking at is how cells attach to this surface and how they move through this system. And they have used some dyes here to dye or to color the cells which are on the surface of these particles. So this, you'll see, I think, later on when you look at your examples, that you have similar types of surfaces. You look at the flux that goes into a surface or out of a surface. And this is a very similar kind of example of a surface where something is hitting this surface, and yeah, you can model that. So what I also wanted to mention is that you need to verify models, and you need a way to measure what's happening. So it's all well and good to develop a whole lot of models, but do they actually mean anything? And that's the actual proof of the pudding, you can say, and is one of the most important parts of the whole thing. If using maths, of using your calculus, making the model, it doesn't matter if it doesn't reflect reality. So you need a way to measure what happens. And if you want to measure what's happening in a complex system, the movement of things, then you need a way to track them. And what I have here is an example. It's called PEPT, positron emission particle tracking, which is a really cool thing, actually. And I'm going to show you a YouTube video here. It's only very short. And I'll just show it, and then I'll comment on it afterwards, because you might not quite realize or understand why I'm showing it to you. Yeah, so of course it's cute and sweet, and I'm hoping it'll make you happy, and I think that this has been a good presentation and get good evaluations. But that's not the whole reason. What we see here, we see this little robot guy, and what he is doing is he's holding something here in his hand, and that's represented here. It's actually being measured by these things. The position of this thing is being measured in real time. So he's wiggling it backwards and forwards, and this device is able to follow it, capture the movement, and plot it in 3 dimensions and represent it on the screen there. So he's got something, he's moving it in real time. And this is a particle which emits positrons. And this weird gadget that's around it is a so-called positron camera. So this is emitting positrons, and these laser lights here, they're like the vectors which are going back away from this particle which is emitting positrons. And they hit this thing on each side, and it measures it. And why am I showing this? This is because it allows you to measure the movement of particles in a system. So it could be particles in a flow field. It could be particles in a piece of equipment. They can look at oil movement in a working engine with this kind of thing. It's really quite a powerful thing. So this is the idea. You have the thing, it emits positrons, they go out, they get detected here, and it gets plotted out in 3 dimensions with the help of vectors and very sophisticated model and mathematical software. This is a picture of the device. So it looks a bit like an MRI, something you'd see in a hospital. And you can also use it for imaging in a hospital environment. There's an example that I'm going to show you now, which we've worked with. So there's a chromatography column here, and this is full of particles. And what we wanted to know is, how do the particles move in this thing? Because it's a fluidized bed. How do they move? And there's one labeled particle in here, and it gets measured in the X, Y, and Z directions. That's what you see here, movement in the X direction, the Y direction, and the Z direction. you take this information and then you can plot it out like this. So here you see our vectors for the movement in the x and y plane, and here in the z and y plane. And of course, you can also get it in the x and z plane. So you see this particle is rotating, and you can see the vectors here and the length of the vectors. And you can take this information and then you can plot it out like this. So this is the column. There's 3 labeled particles at different places, and the flow rate in the bottom has been increased and increased. And you see that the particle stays about the same place in the bottom. Here it's also about the same place. But here there's a lot more movement, a lot more dispersion in the column, which is a bad thing for chromatography, for purification. So I'm almost at the end. You can take that information and then you can plot it in 3 dimensions. So this is a 3-dimensional representation of the same information. So you can take all these vectors and you can plot it out in 3 dimensions, and you can see the way it's moving. So this is just one example, and you can imagine that you can apply this to a whole range of mechanical, biological, whatever problems where you have fluid movement, air movement, gas movement, particle movement in something. And you can use the techniques that you're going to hear about in this course to understand and model and predict it. And that is the end. Thank you very much.